Greetings, everyone, and thank you for joining us for the GROW webinar. It is my pleasure to introduce our speaker today, Manuela Nagel. She studied agricultural sciences at the University of Göttingen in Germany. During her PhD, she developed a strong passion for the long-term preservation of plant genetic resources and conducted research on the quantitative genetics and biochemistry of seed longevity at IPK in Gatersleben, Germany, and at the Millennium Seed Bank in the UK. Since 2016, Manuela has led the Cryo and Stress Biology group at IPK, where she manages the cryobank that preserves around 2,100 potatoes, 250 allium, and 150 mint accessions. Her current research focuses on stress responses and molecular mechanisms during cryopreservation, the role of endophytes, and using genotype-by-sequencing data to support strategic conservation. Manuela shares her expertise as a lecturer at the University of Göttingen. Furthermore, she is actively engaged in advancing conservation science by co-organizing workshops and leading the development of the global strategy for the conservation of potato. The title of her presentation today is Plant Cryopreservation: Principles, Applications, and Challenges of Banking Plant Diversity in Ultra-Low Temperatures. Welcome, Manuela.
So, thank you very much. Thank you very much for this kind introduction. It's also a huge pleasure for me to be here and to speak in front of this audience. I hope you can hear me, and I'm not in front of my slides, so it's a bit different setup. So if you like to see something what I covered, you have a chance to download later the PDFs. Okay, let's start. As already mentioned, I come from the Leibniz Institute of Plant Genetics and Crop Plant Research in Gatersleben, and we house the Federal Ex situ Gene Bank for agricultural and horticultural crops. The gene bank is located here in the middle of the IPK, where we have the seed storage. Actually, we have quite a comprehensive herbarium with more than 500,000 voucher specimens. We have a documentation group, and also my group, the cryopreservation group, is located here. Over the campus, of course, for the gene bank, we have a number of greenhouses, but also fields, and here in the background, a field gene bank. And overall, we have, with this together, 145,000 accessions of orthodox seeds, which are stored at -18 degrees in our cold storage facilities. We have a field gene bank, which is mostly Allium, actually, with 1,380 accessions. There is a slow growth storage with more than 3,130 accessions, mostly potato. And as you heard already, also mostly potato is in cryo. There are 2,500 accessions. So together with my colleagues, actually, I had a chance some time ago, together with Bart Panes from Leuven, Valerie Pence from Cincinnati, Dani Ballesteros from Valencia, Maurizio Lombardi from Florence, and Elena Popova from Moscow, to write a review in the annual reviews on plant biology about the principles of applications and challenges of plant cryopreservation. And we thought it's also a good title for this presentation, and to give you a bit more fundamentals on the plant cryopreservation. So now I'd like to guide you a bit through the structure of my presentation. At first, I would like to talk about importance, come a bit to the history, principle, practice, and challenges. Okay, let's start. Why is plant cryopreservation important? Before I explain the importance for those who haven't heard anything about cryopreservation, so we define usually cryopreservation in a way that's the storage of tissue, of material below -130 degrees. And at this storage temperature, at this ultra low temperature, you usually have a very minimum or even no metabolic activity. And so everything is arrested, and therefore we can have a long-term storage, because so far we don't know how long the storage can last, so it's more or less unlimited. So what we store, why is it important? Of course, for me as a gene bank manager, the conservation of genetic diversity is the most important issue, and specifically of clonal crops, because I'm here in a crop gene bank. And we store specifically those crops which either do not produce any, plants which do not produce any seeds, for example, like garlic or like banana, which do have few seeds. This is the case for mint. For recalcitrant seeds, we don't have any here, but quite a number of fruit trees are recalcitrant, or for those plants which do not breed true and you cannot maintain the variety via the seeds, which is the case for potato, for example. So the same facts become true for woody species. They also need maintenance, like apple trees. Quite a number of tropical plants, of tropical fruit trees, produce recalcitrant seeds. And the only way to long-term store them is cryopreservation, actually. Another point are the wild species. We know that our environment is changing due to climate change, due to loss of habitats. And here we have quite a number of wild species which are predicted to be lost in future. And here we need to do something. And also industrial plant cell lines, which are important for the industry and to produce some compounds. However, cryopreservation is not only important for conservation purposes, it's also interesting for plant breeding and production, actually. And for example, we had some time ago here a small Christmas tree collection, because if these breeders cross trees, they get a lot of diversity, but they cannot test everything at once. So they cryopreserve their clones first, and then they test them after some time, after they have everything tested, and then it goes off cryopreservation. Another issue is also virus eradication. So quite a number of viruses can be eliminated by cryotherapy, and which is also interesting for production, of course. So these are the fields which are important for cryopreservation. However, you can maintain these plants also in a different way. Of course, they can be maintained in the field, which is actually the traditional method, and where you have the possibility to see the plants in nature, to phenotype them, to provide material. But you have the disadvantage in the field that you have accumulations of endophyte and mutations. You have exposure to adverse weather conditions or pathogens and insects, and even plant aging. So that means that quite often the plants need to be transferred into a new field every year. So about the risk, the researcher became quite aware of that, and it started already in the 80s that we started to back up these field collection in vitro. And therefore, in vitro slow growth storage has been invented and developed. Slow growth storage means that usually we store at low temperature and low light conditions and try to extend the vegetation period longer than normal. And for us, we have, for example, some mint in our slow growth storage. We started with this material in 1997, and you can see it's there since nowadays. Of course, we need to renew this material every 2 years. So we have between 2 and 6 degrees storage with low light, and the mint accessions behave quite nice. So they do not have any problems. If they become old, we transfer them to a new medium, and then it moves on. We have a different case for Allium, actually for garlic and also some onions. And here you can see that my predecessor, actually Joachim Keller, he introduced a lot of Allium species into our in vitro storage. And but here we realized quite quickly that this is not so easy. These plants age very fast. You cannot multiply them after some time. It's very hard that you get new plants out of that. And then they also produce or accumulate more endophytes and have contaminations. So, and at the end, you had to transfer them in the greenhouse and back. And due to this workload, it was finally decided to not have an in vitro collection on Allium plants anymore. So when we have in vitro plants on Allium, it's only for cryopreservation, but not for the slow growth storage on its own. So that means also the in vitro slow growth storage unfortunately has some problems. As mentioned already, in vitro calcitrans, that's the issue with Allium. Plant aging, we can have here also soma clonal variations. And for some species, you need really specific protocols, and you have infestation. You can have infestations of insects and contaminations. So the only approach where you can really store your material over long term, at least the clonal material, is in cryo. And here you have a high genetic stability at a minimum space requirement. Over long term, costs are also low, but just over long term, we come back to this number later. And you have so far unlimited storage, we usually say about 100 years. Yeah, and that we need cryopreservation is quite clear, because when we check the different papers and survey which have been done, so we calculated that there are collection of more than 400,000 accessions. Probably it's even more, because some gene banks didn't contribute to this survey and we didn't catch. And only 33,000 are at the moment backed up in vitro or just maintained in vitro. That could also add up. And only half of the in vitro plants, or only 70,000 or 18,000, are at the moment in cryobanks safely stored, that the genetic resources are really safe. So that's actually a very low number, and we need more cryopreservation. Yeah, there are some institutions who are doing that quite for a long time. So specifically USDA, the International Potato Center also, where of course has all its potato collection duplicated. We have here the Biodiversity International in Leuven with the banana backup. We also have the potatoes and the Allium species. In Japan, we have mulberry and potato, and then we have in South Korea garlic and potato. And you can see a lot of greenness here. So green is potato. So that means specifically one of the most important food crop is, of course, so far in cryo, also some red, the apple are somehow preserved, and Allium species, but other species are really behind the storage, and we need to do more, and specifically when we talk about the wild species. And we have not really numbers about King's Park, for example, how many species, how many accessions, or South Africa are stored. And there we need more surveys and probably also more funding for the cryopreservation in general. However, coming first to the history, how did it start that we have cryopreservation? Because this is one of the youngest conservation approach. And so what's the historic background for that? And it all starts with liquid nitrogen, because that's very essential. I mean, you can have also minus 150-degree freezers. However, liquid nitrogen is, in terms of cost, probably cheaper. And the element on its own was discovered in 1772. It's relatively safe because we have an inert gas, colorless, odorless, non-corrosive, non-flammable, very cold and cheap. And we know it's very cold, that's a danger. But the good thing, it's in the air. So there is no issue with any toxicity. However, you should make sure that composition stays the same. If we have too much nitrogen in the air, it could be also harmful. So if we have 78% in the air, it boils at minus 196 degrees. What's the good thing for us? Freezes even at minus 210. And cryopreservation would not be possible if we wouldn't have the liquid form. And this started in 1883, actually, with Wroblewski and Olszewski, who discovered or who invented the method how to liquefy nitrogen by compressing and cooling of air. But we just have liquid nitrogen, which evaporates very quickly. So now we needed a method to store it. And that was actually 1898, James Dewar, who invented these Dewar vessels here with double walls and the vacuum in between. And he was able to really preserve or maintain this cold liquid for some time. And without this invention, we wouldn't be possible to store it. So that was the initial start. And then with the beginning of the 20th century, of course, quite a number of researchers started to work on the cryopreservation, at first in human cells, in blood cells, and in sperm cells. And here it was around 49 that they discovered that there are some cryoprotectants which help during storage. So glycerol and DMSO were quite quickly found. And the first human cryobank then opened in 1943. But you can see here, plants, 1960. So we obviously, the plant side, took a moment longer. The reason is plant cells are very stiff. We have the cell walls, we have the vacuoles with a lot of water inside, we have the chloroplasts, which produce a lot of electrons, which are also then probably harmful for the process. And therefore, it took a while until the method really could be developed. And it was a Japanese researcher, and I'm very sorry for this bad picture quality here. It was Akira Sakai who started in the 1960s to do experiment on mulberry trees, actually, on winter buds. And he realized that if we choose the winter buds, and there is no pretreatment at all, and puts it into liquid nitrogen, there's also no survival. However, as soon he is changing the pretreatment to lowering slowly the temperature down, there was an increase of the survival after liquid nitrogen treatment. So, and by doing these experiments, he actually realized how winter buds survived the cold temperatures in our winters. And to visualize that a bit more easier for you, I put here a plant cell. And I mean, you can imagine that when a winter bud, when a branch, a tree is exposed to lower temperature, cold acclimation starts. So the tree starts to develop some protective compounds when it's at lower temperature. And then at a certain moment, it starts to freeze. And here we have done the phenomenon that we have a so-called supercooling. That means at temperature below zero, we do not have ice crystal development. That means in the cytoplasm here inside the cells, there will be no ice crystals until minus 10 degrees. But as it's cold outside, so the ice crystals start to be in the intercellular room. And because you have here now ice crystals, the water potential drops and the water follows the water potential, so that we have here a moving of the water outside the cell, so that we have here a reversible plasmolysis, so that more water moves out, and we have an increase in concentration of the cellular solids because we have more sugar then, and we have more protective compounds, for example, and this helps to avoid further ice crystallization inside the cells. However, every system has its threshold. So if it goes too far, if the pre-treatments are not good enough, then of course it can also change the system. We have a drop in the pH, precipitation of electrolytes, and then also inside the cells, free water can convert into ice. And this is actually lethal. If we have intercellular ice crystallization, at least at a higher amount, usually this is extremely harmful. Yeah, and then you have that some ice crystals go together to huge ice crystals. So that are the principles how winter buds survive the winter, at least until this point here. And based on that, it's still done until today, actually, that the dormant buds are sampled in the midwinter when there was already cold acclimation. Then there are some cuttings into smaller pieces, which are also further air dehydrated at -5 degrees and until 22% water content actually. And then we have a very slow cooling until -30 degrees to get the water out, actually, to increase the intercellular solution. And then it's thrown into liquid nitrogen, actually, and can be stored. And afterwards, that's the nice thing with spud, quite often you can transfer it directly in the field back by grafting, so that this method can be cheap if you are able to avoid any in vitro steps, let's say. So this is a bit the background for cryopreservation. So what are nowadays the principles? And so how to conserve the different organs in a viable state. And I like to explain it at first on seeds because I think it's a more simple system. So when the seed is not mature, at a very high water content, we considered it in a so-called liquid state, let's say. However, during maturation drying, it goes into a so-called amorphous glassy solid. However, we have quite a number of different terms for the state. It can be also vitrified state, glassy state, or extremely viscous liquid. So they all describe more or less the same state of the material. The important thing is it's not a crystal, it's still flexible, it doesn't increase in volume, and it's able to interrupt then also ice crystallization. What's the huge benefit? And when we move into this state, we move across so-called 5 hydration levels, from the highest one from the liquid state with 99% relative humidity and a very high water potential with -0.5 megapascal. And then cells and organelles shrink, increase in viscosity, at least in the cellular solution. And then we move further down to the hydration level 2, which is actually the so-called glassy state, which we arrive at 50% relative humidity at 20 degrees. And here we have a lower water potential of -100 megapascal. However, if you move this too far, you can come to a very dangerous situation, which is a porous glass. And this can be negative because you lose some protective layers of water molecules. And the good thing of a glass is that you have the reduction in bacteria growth and yeast growth and enzymatic activity and non-enzymatic browning. And however, you have here an increase in lipid oxidation, which increases the reactant, which increases the concentration of reactant molecules. So this is the case for seeds. They are under room temperature conditions in the glassy state. So, and when I try to describe you this on a thermogram, so we are at room temperature here, and 0 degrees is here. this is the temperature where we know that usually ice crystallize. Then at minus 40 degrees, we have the so-called ice nucleation temperature. This means in the space here between 0 and minus 40 degrees, we have a room where the liquids can be super cool. This means they can still be liquid without crystallizing, but if there is a certain motion or a certain energy, they can immediately be transformed into crystallization, but the chance is good that they are not. And then we have this for sure area of crystallization between minus 40 and 100 minus 40. And then here there is a so-called glass transition temperature. Here we have the glass. So this is our safe storage zone where we want to come. And in seeds, we just get rid of the water, probably also some protectant molecules which are developed during drying. And during this air drying approach, we increase the concentration of cellular solids. And with this, the concentration of glass transition temperature increases and the freezing temperature also drops. And with this, we can safely store the material easily, either at minus 18 degrees, which is common, but you can throw also quite a number of orthodox seeds just simply in the liquid nitrogen, and they will also withstand that. And the same is true also for non-orthodox pollen, or also for embryonic exit of non-orthodox seeds. This is air drying. When we move now to cold acclimation and slow cooling, we already touched this a bit with the dormant winter bud. Here we know already that the dormant winter bud cold acclimates, so they increase the number of protective molecules. With that, they also again increase their solids, there's cellular solid concentration, and then we have the additional effect of the slow cooling. And during this slow cooling, we have the effect that we have the very slow temperature decrease, and with this we have the possibility to move here along this zone of supercooling, and at the end we drop the material into liquid nitrogen when the cellular solid concentration is relatively high, so that this is also safe. And we have here this approach for dormant winter buds, also for quite a number of dormant buds. It was adapted for unorganized cells, for embryonic cells, and for cell lines. It's nowadays quite normal to use this method. So, however, we didn't talk about herbaceous plant species so far. So we talked about dormant winter buds, seeds, pollen, and unorganized cells, but herbaceous plant species were quite of a problem for some time. And here the permeating molecules and the osmotic dehydration was a game changer, specifically the combination of these both. As permeating molecules, we define glycerol, for example, dimethyl sulfoxide, ethylene glycol, which enter into the cells and increase again the cellular solid concentration, and the osmotic dehydration, this sucrose, glucose, polyethylene glycol, they also enter the cells, but they are not immediately enter the cells. They probably need to be actively transported, lead to osmosis, and therefore here the water goes out. And as soon, so we increase the cellular solid concentration, and as soon we drop this in liquid nitrogen, we have the vitrified stable storage state. As said, it was the combination of these different molecules which allowed to store shoot tip and also embryonic axis of recalcitrant seed. And it was again actually Sakai who developed this method. And the first one, he started in 1990 to work on the so-called plant vitrification solution 2, which is made of sucrose, glycerol, ethylene glycol, and DMSO. And this is actually also the most common cryoprotectant because it works very easily for a lot of different plant species. For more sensitive plant species, PBS3, he also developed, and this is 50% sucrose and glycerol, and we focus actually on this cryoprotectant because it works nicely for all of our species. And he continued then and until high age even with PBS4 made also of sucrose, glycerol, ethylene glycol. However, it doesn't mean you need always this combination. For example, we worked for a long time on DMSO droplet freezing. That was also working. And for some species, the single compounds are better. But you need to try and to test what's the best for you. And if you may have wondered why you do not simply drop this material in liquid nitrogen, because there is the glass transition temperature here, right? And if we come to minus 190-60, there is a glass. Why not to drop it immediately? The thing is, water content is very high. And we have the problem that under normal conditions, water doesn't go into a glassy state. Only under very specific conditions of physicians, it does vitrify. So what happens is that you may have a lot of small ice crystals, but they also combine. And then at the end, you have intracellular ice crystallization, and this is lethal. So when you drop it in liquid nitrogen, you usually have this ice crystallization, and the plant doesn't survive. That are the major principles how we store nowadays our material. And now I would like to come to give you some examples how we are working with cryopreservation, because we do cryopreserve shoot tips here at IPK, majorly on potato and allium species. And as I mentioned already, we use PBS3 droplet vitrification, at least for potato. For allium, it's just vitrification. And in case of potato, we have in vitro plants, which we use. And in case of allium species, we use field propagules. We excise that. And then the procedure starts. We have a pre-culture treatment where we increase already the solute concentration by adding 10% sucrose. Then we have a so-called loading with another 12% sucrose and 12% glycerol. And then we have the final cryoprotectant, PBS3, which is 50% sucrose and 50% glycerol. And after 2 hours with this treatment, we produce droplets for the potato or the garlics, which are very heavy shoots. They go directly into the vials and they are thrown into liquid nitrogen. Yeah, and then either we rewarm them because we want to know, is that really, was this procedure successful, or store the material. And here you can see Marion Grübe. She's doing the potato cryopreservation here at IPK. And she did plant the material probably 4 to 6 weeks ago. At first under normal conditions, then there was a cold hardening. And here she extracts now the final in vitro plant. So you see she removes the upper leaves, that she only has a bit of the meristeme and some small parts of the leaves left. Then it went into a sucrose solution, stayed overnight. So we shortcut the video here. And that was the process when she moved that into the droplets and then into liquid nitrogen. So, however, we do cryopreservation since 1997. And of course, we do the adaptation during the routine cryopreservation. And as said, it started with DMSO droplet freezing at first. And this is an increase of the collection, which you can see We received material from the Western German gene bank, so which increased then to 800 accessions, and then we started to play around with acclimation temperatures. And nowadays we still use 5 degrees in dark condition up to 3 weeks. We started to use microtubers as basis multiplication source because it seemed to have less endophytic contamination. We started to have a backup in Braunschweig, so we have our security backup of all the material. And in 2016, we started PVS3 droplet vitrification, so we changed the method. I come back to this a bit later. Nowadays, we use a bit less sucrose in the medium and direct cooling, always just to improve the regrowth after cryo and try to help to support the long-term storage. Nowadays, we have triplicates also. We have 2 tanks here and 1 in Braunschweig, QR codes, and in the safety backup, we use also this pickle film from Gene Everett to print our passport data on that to have safe information along with the cryo samples. Yeah, these are about 2,133 potato accessions at the moment. However, of course, I mean, nothing is ideal. We also do have challenges with regrowth because sometimes they have contaminations, and that's overall our accessions, also those ones which are not considered as securely safe. And overall, we have a regrowth of 52%. We just recently changed our threshold level from 50 to 30, just to be sure over a long time that we have a safe storage method, and just need to redo the cryopreservation for those ones which have a lower level. Then we have also Allium cryopreservation, and here we use field proper goods. That's a bit different because here you can, for example, use pollen. This is directly accessible in our field, dry and store. You have emerger inflorescences, for example, bulbils or mature inflorescences, where you can prepare and extract directly the shoot tips out of that. You can also get the shoot tips out of the cloves. Or you can use the meristem from the basal plate or put the whole thing in vitro and use the in vitro plants for cryopreservation. And you have a range of material available for the cryopreservation. And here I want to give you a glimpse on the cryopreservation or on the accession of shallots. So we harvest usually this material in July, then it stores for some time to get rid of dormancy, and then we cut the cubes. Here in this case, my colleague Sibylle Freist, and then she starts to sterilize, of course, this material. And the nice thing with shallot is that you have quite a number of meristem inside, so you can get more than just one. This is different in garlic. And then you peel. And for those who have done bananas, it's very familiar, I think. It's very similar because it's a monocot. And you peel until you come to the meristem. It takes some time, and this is also very meditative, actually. And then you go down to the state that you have more or less one meristem and the leaf which covers the meristem. And with this, when this is exposed to the cryoprotection, this usually survives quite nicely. However, here we have cloves, bulbils, in vitro plant inflorescences in our cryo with 224 accessions. Here we have the same variation, and on the average, the results are even lower. The problem is the same as in vitro. We have here higher contamination, specifically because it's field material what we have here. And yeah, also we do have the problem that we do not have so many seed material, field material, which we would need for our research. So we use also a lower number of explants. So Allium is difficult in vitro, but also in cryo. So, and with this, I think I almost started to talk about challenges, because Allium recalcitrancy and endophytic contaminations or contaminations in general, also fungi are an issue. And but first of all, the challenges which I actually see is the cost component, especially for gene banks who just like to start with cryopreservation. I mean, we really need to store more in cryo to protect our diversity. However, as soon as you need to go via in vitro, you of course have to have well-equipped labs, sterile working conditions, you need trained staff, and this of course increases the cost. And we had here in Gatersleben 2030 some calculations that when we maintain the material in the field, 47 euro would be the annual cost for the field maintenance of an accession. When we introduce it to cryo, dependent if it's a vitro or a field material, it's between 360 and 560. So that would be probably possible to do. But tried to do an estimation for banana in 2020 with 900 euros in vitro and 1,300 euro for the cryo, the introduction into cryo. So the first cost which you need to introduce this material, and then it becomes cheaper, right? Because then you have just the liquid nitrogen cost, which be probably 1 or 2 euro per accessions over a year, and then you have after 15 or 20 years amortization of the costs. However, the introduction is relatively expensive, and actually when I consider potato nowadays, because we need to go via sterile cultures, we multiply relatively high number of plant. I would even say that it's in the 1,000 after Corona and all the crises we have now. I would estimate it's between 3,000 and 5,000 euros. We haven't done a real good cost estimation, but it's expensive. And over time, this cost will be definitely lower, but it shows that it's really a backup method and not really a method which we can bring something into cryo, take something out, bring it in. So that's not a method for active storage. It's really a backup storage. So then, because it's so expensive, of course, we need to know what is duplicates. What are duplicates? Because we don't want to cryopreserve the thing several times. And here, I know that the international gene banks are doing that very regularly. They investigate, they genotype their material to have not too much in the storage. And we started to do this also with our field material. Our PhD student, Song Wu, has done the GBS analysis of our material here in the field. That's garlic. And these are very preliminary data, I must say. And we estimate that between 10 and 50% of this material is probably potential duplicates. Now it comes that we need to verify, which is also a critical step. And we think, for example, that there seem to be 2 duplicates, for example, from reddish skin garlic accessions, which comes from Italy and entered the gene bank in 1981. And then there's also a reddish skinned garlic, which was called Italian purple, a few years later, which came from the US. And we assumed that it could be easily transferred from Italy to the US, and then we got it back and have it as a duplicate. And we would love to prioritize at first those ones which are really unicates. That would be important specifically for garlic. I touched this topic already. Number 3, the challenges are contaminations and endophytic organisms. And we have this problem quite regularly. And here, with a team of collaborators, we were able to barcode 385 accessions to understand a bit more about the microbiome of the material. And we found quite a number of microorganisms. So this is material what was in storage, either with very high germination or regrowth, or very low, which goes to 0, actually. And we expected that the composition of this microorganism would have been different, that you clearly see that some of these microorganisms maybe have a harmful effect, where others have a good effect. We are not sure anymore. I mean, for the high ones, you see there is a different kind of composition for the high and low ones, also for the medium ones. But at the moment, it's hard to estimate if there are really some which have a harmful effect on that. And yeah, this research needs to go on to see if we clearly can identify maybe also some endophytes which can help cryopreservation. Let's see. Then there is another thing. Of course, you have to develop for each plant species the protocol. Some of the plant species you can just use a protocol which is available, try to transfer this, and then it's working. For some plant species, that's not possible. And here, when we switched from DMSO to PVS3, fortunately, it was easily working. And we used the protocol actually for garlic. We just prepared droplets. And we saw then also in the different potato varieties that the PVS3 droplet vitrification increased the regrowth compared to the DMSO droplet freezing. And that was the reason to switch. However, we tried also a bit to maybe optimize, because we wanted to be faster, because the DMSO droplet freezing has a pre-culture of 20 hours. Also, the PVS3 droplet vitrification has a pre-culture of 20 hours. You need to wait overnight. And I knew in PVS2, it's possible to proceed immediately. You don't need to wait overnight, you just wait in half an hour. So we assumed maybe we can try that also for PVS3, but some other people, those who developed the protocol, realized already that's not possible. To reduce that to 30 minutes pre-culture is too short. And you see it here, that's the gray one, or that are here different accessions which we try, and all the gray ones have very low survival and regrowth, whereas the PVS3, the normal ones, very high. But this experiment gave us the idea that you need the sucrose, and you need to expose it to the sucrose for a longer time when you use PVS3 droplet vitrification, because probably the sucrose need to be actively transferred into the cells and work also as in this way as a cryoprotectant. It's not enough when it just stays 30 minutes in the solution. And then another method which my colleague Anton Petersen and Dr. Olena Kishenko just recently worked on. They developed a protocol for duckweeds, also using PVS3. And they found a very fascinating phenomenon, that the light has a huge compound. I mean, we know it before that light is very important, but here we have, for example, green cultures. So this material is developed under normal light and then cryopreserved, whereas these cultures, these are etiolated cultures, were not exposed to any light. But if you compare that, here we have immediate growth of the material, and here these plants were developed under light, and then for a few days they were exposed in the darkness, and these plants were also a bit faster. So here, no light in the pre-culture helps a lot with the regrowth after cryo, and also the light after cryopreservation is important. There are many factors, and it's very hard to list all of them, but for those who went into new species, it's quite often very hard to develop a new protocol. You have to take in mind the growth conditions with temperature, light, medium composition, insects, infections, genetic setup, for example. Protective compounds are important, tolerances against stress, and then the cryogenic temperature with cold acclimation, mechanical wounding, osmotic pressure, toxicity, and cooling procedure. And there are many factors which can influence. Yeah, then there are also further challenges. For example, cryopreservation is slow quite often, at least for shoe tips. You need the excision step, you need the procedure. We are able to do about 100 accessions per year. For me, epigenetic effects need to be further investigated. It's still not very good developed. We need more standardization and data security. Actually, there are some new rules on of the FAO coming, but they give a lot of freedom. It would be nice to have more discussions about standards. Backup storage is, of course, very important. I mean, if you have a fire in the institute and just one tank, all your work is lost, actually. And you should also keep in mind the cryopreservation is very important as a backup method, but you need also the complementary approaches. You need the species in vitro or in the wild that they can be used and they can be phenotyped. And with this, I'm coming to an end. I hope not too long. I'm not too long over the time. I brought you from the importance of cryo to the history, some principle, practice, and challenges. And I hope you got an idea how cryopreservation works, who is doing that, and who you may approach in future of the different cryobanks. With this, of course, I like to thank to my team who are doing all the work, doing the standard cryopreservation, and of course also the scientists who are working on the different methods together with the team in the gene banks who is taking care for the material in the field, also documentation group, and other groups at IPK who support this research, and yeah, different funders. And then I'd like to thank you for your attention. Thank you very much.
Thank you, Manuela. That was great. Thank you for walking us through all these aspects that gene banks must consider if they want to go into cryopreservation, which is on the top of many national banks. So we have 3 questions, and I'd like to ask you these one by one. The first one is there a DNA sequencing technique that can be used to support cryopreservation?
So what we did so far is when we developed the dark wheat cryopreservation, we checked if our number of chromosomes stayed stable, but I doubt that there is a change in the DNA. If there is something changes, is it more on the epigenetic level, the methylation levels, that these change? And there is not a standard at the moment. There's bisulfite sequencing, which you can use, for example, which is at a high throughput level, but there are also single markers I think you could use. But more the high throughput technology is the more standard one, and I would suggest also to use this one because it gives more results and more ideas. I mean, we have done all these different methods, and we know there are some changes here and there, but we need more a better picture of epigenetic changes, and I doubt that there are DNA changes actually.
Wonderful. Yeah. Our second question, you've already talked about it, but perhaps you can expand a little bit more. And it's, can we preserve seeds under cryopreservation?
Yes, so definitely. I know that the USDA is doing that quite commonly. So they test if the orthodox seeds, if they dried well, they just expose it to liquid nitrogen and then store also part in their cryo storage. So this material is well preserved. However, what we don't know, I mean, there are many studies who investigate if you can immediately thaw this material and if it survives, but we don't have really experience on the long-term storage. If it's better than -80 or not, which seed moisture contents are required. So I think there is a bit a lack of study, which would be still helpful for real standard seed cryopreservation. However, for the short-lived seeds, would be very nice to have.
Yeah. The third one is about an alternate methodology, and it is, did you try cryoplate method? It increases the efficiency of cryopreservation in many crops, such as potato.
Yeah, I'm aware of this. I mean, in Japan, they developed this method and many researchers use that. We didn't see for us a beneficial effect. I think it's very nice in the handling, um, but it didn't really give a huge benefit for us that we used it at the end. But I guess for some new species, and especially also for people who start with cryopreservation, it's a nice way to standardize that.
Okay, going back to related to seed preservation, the question is cryopreservations of seeds or seedlings, does it make sense? And thank you for a great presentation.
Yeah, that depends on which type of seeds you have. If you have orthodox seeds, just throw it directly in liquid nitrogen and see if it survives. That's probably easiest and fastest. If you have a recalcitrant seed, use the embryonic axis if this is possible. Then seedlings is probably also possible, but the embryonic axis are, I guess, more in a already denser state with lower moisture content. And so I would not go for seedlings, I think.
Okay. This one has to do about records of storing. And the question is, do you know which plant was stored the longest? And if it's published somewhere. By stored, I don't know if he or she means cryopreserved the longest or, something like that. Do you know of the longest cryopreserved plant? If it is in fact cryopreserved.
Yeah, so we have 1997. I think that's already quite long. I would assume that the USDA has some material, but not shoot tips, probably more dormant buds, which are stored longer. Also, garlic in South Korea might have a longer storage history. So there are some collections, but I never heard about the longest stored accessions in cryo. This I never read about, but I would be excited to see that. So if you have a reference on that, I would be happy if you send it to me.
Yeah. And the last one, it's more a comment, and it's about the cost. Cost should be, actually we have another one coming in, but let's take this one first. Cost must be calculated on a turn basis, like 20 years comparison. So 20 years in storage, cryopreserved versus 20 years preserved in the field. And that might be, according to this person, a good comparison, in time, rather than a one-time thing saying, okay, to introduce into cryo, it would be this much. But then what about maintaining it? And so that's simply the comment, and wondered if you had a response to that, a look at 20 years of field versus cryopreservation costs.
Yeah, I agree. I mean, at the end, that's what's count. The long-term run, we have the lower costs in cryo. So definitely you need to take to account the storage period. However, I think when you especially start with cryopreservation, I wanted to provide these numbers because you should be aware that the initial costs can be extremely high. This you have to know, I think, but it will go down definitely over long term, and that's the benefit versus field conservation. Yeah, I agree.
Yeah, and related to that, actually, they're wondering which method is better, whether field gene bank or cryopreservation. And you've already talked about that, but can you say it just a little bit more to answer that comment?
Yeah, I mean, the field has a huge benefit that you have the plants there. You can phenotype, you can use it, you can give it easily to somebody else. If you have it at cryo, you are sure it's there. You do not lose it because it's in the field and is exposed to any worse weather conditions. But in cryo, of course, you cannot use it. You have to rewarm it. In terms of potato, I would estimate that until you get new plants, very nice plants, 2 years maybe. I mean, in vitro develops faster, but I would assume 2 years until you have somehow like a normal plant, and here you cannot easily use it. So both methods have their pro and cons, and you really should consider this as complementary approach.
Okay, we have two more comments, and that is about native plants. Is there a program for cryopreservation of native plants? Also seeds and seedlings, but you've already addressed that, so it would be about the native plants.
Yeah, there are too few activities. I know that Valerie Pence from the Cincinnati Zoo, she's actively trying to organize symposia online, also meetings. So that's very active. And she also tried to get fundings to preserve native plants. In which scale and which amount of money is there, I have no clue. Also in Australia, there are some programs. In South Africa, but you don't really find a lot of data on that. And it would be nice if it's more organized, similar to the clonal cropped species, actually.
Okay. And our last question is, can you integrate NGS to detect indel variants? Could they be detected after long-term conservation?
Next generation sequencing. So to get indels, so I didn't get the question
Yeah, to
exactly.
detect any indels, INDELS variants uh,
Mhm.
that can be, and can they be detected after long-term conservation?
I didn't try, so we didn't follow this. So maybe it's interesting, yeah.
Okay. I think with that, I'd like to thank all the participants for hanging in there. We had wonderful questions. Thank you so much. And also thank you, Manuela, for putting a wonderful presentation together. That was really, really in-depth, and I like the many aspects. I think we were lacking this sort of cohesive information for all the challenges and the principles and the history behind that. So last words to you.
Yeah, of course, to my heart, it's very important that we need to make sure that cryopreservation is going on, that we can back up all these clonal varieties in the fields, but also wild species, and therefore all the different types of fundings and projects need to be supported. And I think that they just actually started a very nice project from the ECPGR, which supports this cryo storage in the south of Europe. And that's a great opportunity, and more of these possibilities should happen. So it's more a question and to the funders to go for more cryopreservation.
Thank you very much. Thank you very much, everybody. See you on the next one.